Hi, welcome back. I hope you had a nice spring break. This is a little bit different this week. I'm going to skip ahead to chapter 20 and do a little bit of chapter 20 to help us where we are going. We'll come back to chapter 20 at the end of the class as we get closer into understanding the modern way of looking at DNA and genetics. But for now, I want to introduce some of the things we're going to do in lab for the next couple weeks. So before you watch these videos, review the pGLOW idea and what we did in pGLOW and horizontal gene transfer, and basically how we control for gene expression. How do you know so far that we turn genes on or off? This is Alice. I'm going to try to scooch her away. I have less singing during our genetics, but who knows? It's early in the morning for me. We are going to do chapters 14 through 19, and chapter 14 is part of this week's assignments. But for the next few labs, this information is going to help you get through them. So for big ideas here, we're just doing part of chapter 20, and in the homework, I just assigned a little bit of chapter 20 PCR explanations, and then a little bit of a review of chapter 10 when we talked about electrophoresis. Your job is to, in this lecture, figure out what restriction enzymes are and identify them. We will be using 3 in lab. I will introduce them to you this week, but they're also part of this lecture. Explain to me, again, how bacterial plasmid vectors are used. That's what we inserted into the pGLOW bacteria experiment. Talk very briefly about the concept of a genomic library. We're going to look at the actual function and how PCR is used and how it actually works before we do it in the lab. I would like you, as going forward, to compare PCR to DNA replication. I know I talked about it, 2 weeks ago, but the idea that those things, PCR is replicating DNA replication in the lab, and then a little bit of a review of gel electrophoresis. All right, so the idea of recombinant DNA is going to tweak some things in your head. We talked about recombination when we talked about different alleles coming together when you inherit one from one parent and one from another, or you go through crossing over. We talked about the concept that we could recombine different alleles so that maybe change the ultimate expression that was made by an organism with recombined alleles. This is a little bit different, but same idea. The idea that DNA can be artificially brought in, so instead of sexual reproduction or horizontal gene transfer, I can do it in the lab and I can recombine DNA from one organism to another organism and change the expression of types of proteins that the new modified organism can make. There are 2 major tools that we're going to introduce today that do this. One is restriction enzymes, which is really the concept we've already been here, but we just haven't named and described them as much. And then cloning vectors, which is also we've done a little bit, and I want to show you and build it into the concepts of genetics. So restriction enzymes were first introduced or understood in the 1970s. Dana and Nathans actually published a paper talking about an enzyme in bacteria that cuts DNA at specific locations. This is an enzyme that many bacteria already have inside of them. Its job is basically to identify plasmid or viral DNAs that don't belong and snip them up at various locations so that they're no longer functional. This is part of the bacteria's defense mechanism against different viruses. So it's something common in bacteria, but it's not the way that we as eukaryotes or humans even fight off bacteria or viral infections inside of our bodies. This enzyme that they recognized has been seen in a lot of different bacteria. We classify them as restriction site enzymes or restriction enzymes. They find a specific code on the DNA of the virus and cut the DNA there. When they do this, what we have been able to do in the lab is take those restriction enzymes and identify the location where they cut, and then use that as the first step of then adding in a piece of DNA that we want to add into it or genetically modifying this organism. We're going to use a couple of these. Hindi3 is one of the ones that we're going to use in the lab. What these restriction enzymes do is they find a specific sequence and cut at a specific location. They're like molecular scissors. Notice here that HindD3, this sequence is AAGCTT. If I look at the other side, the complement of that on the DNA, it's AAGCTT. These are reverses of each other. They're palindromes. So when I look at these two sequences, the restriction enzyme cuts here between the A and the A of the sequence TTCGA. And on this side, it cuts at the same place, TTCGA, cut. This creates two sort of pieces that stick out from either side of the DNA. The original function of this in the bacteria would be to cut it, and now this gene that it interrupted is no longer functional. The virus doesn't work anymore. But what we are going to do then is insert our own piece of DNA here and our own piece of DNA here and add in functionality to this. We can buy about 600 different varieties of restriction enzymes now. So commercially available means you can go on the internet or go to a company and order them. And these are places where we practically insert most of our genes, but we'll actually know many more of them. These are very common among the bacteria. The restriction sites are absolutely distributed randomly through a genome. Remember, and I want to keep bringing this idea back. We can't see this. So when I am bringing in my restriction enzymes, I know that I am cutting at that sequence, but I don't know where that sequence is in the thing that I am cutting because I'm really imagining the sequence of the DNA, but I can't actually see it with my eyes. We'll come back and talk about how we can see it with our eyes, but that's not a simple process. These things are really, really small. We do know that the restriction enzymes looks for palindromes. Palindromes is when it reads one way and the opposite side, when it reads one way and the opposite side is finding the same exact sequence. So again, here in HindD3, it's looking for AAGCTT, AAGCTT, the same sequence in opposite directions of each other, and it's able to cut them. Most sequences that the restriction enzymes are looking for are somewhere between 6 to 8 nucleotides long. I want to point out a difference here as well as we go forward. Most of the enzymes that we're going to use are going to cut in this way, where they leave these cohesive ends. They kind of leave where it's laying over. And if you've ever, I don't know, even in a craft kind of, or in woodworking, tried to put two pieces of wood together, it's super helpful structurally wise for you to have those little overhangs so that it holds the molecule or the wood together a little bit better. Some restriction enzymes do cut with blunt ends. This is looking for AG here, TC, TC, and it cuts right down the middle. These blunt ends are usable, but they're generally, for our purposes in our lab, a little less cohesive. I'm going to have to put them together in a different way because I cannot rely on sealing this part together by using hydrogen bonds. When I insert my new DNA, it's going to be AGCT here. And I'm going to rely on the fact that these fellows will form hydrogen bonds to start to stabilize the molecule before I put my phosphodiester, the covalent bonds, in place. Here, I'm going to need to seal it with here to here covalent bonds. So I'm hoping after we have talked a little bit about the structure of DNA and DNA replication, and you've definitely done transcription by this point, I'm hoping that a lot of these things make sense to you as I start describing the lab processes of them. If not, 100%, talk to me in lab. We have this week, we have a little bit, one day of setup and then one day of actually watching the experiment happen. But I have time before and after the actual doing of the lab, and even during it, because once again, we're going to wait for agars to cool, the most exciting thing, watching the thermometer drop. Reach out to me. Let's see if we can make sure that this makes sense to you, because it's really building on everything we've learned so far. One of the first identified restriction enzymes was from E. coli. So it's called EcoRI. It's strain R, which is the type of E. coli that it is. This may be eco-re. I am working with the disadvantage of I only know the way that my professor called it. And I have realized as I have listened to videos over the last, 10 years, that he was making up how to say things. I think this is eco-RI. Just in case. I didn't check this one. When I take eco-RI and I use it on a strain of bacteria or a strain of DNA, where it cuts here and here, these are palindromes of each other, AATTC, AATTC. And if you're not looking, I'm going in opposite directions. It cleaves at the location on the DNA where the restriction enzyme binds to the DNA and then cuts, like molecular scissors. I have two different fragments here. This is from one source and a second source. Hi. My goal is to put these two together. So what I do, and I'm kind of moving down the picture here, is I have this little place where I have a nick, a cut. I have another place here where I have a little cut. And then I put my two different sourced pieces of DNA together and they form hydrogen bonds. I allow them to anneal. And we have defined anneal in lab, but I don't think I gave it to you as a vocab word in lecture. Anneal means when I put two sticky ends together. In PCR, we're going to do this by changing the temperature to something a little bit cooler there where we started. It's going to cause hydrogen bonds to form. These hydrogen bonds are strong enough to hold these two fragments together, but we're not done, just kind of like we're not done when we form hydrogen bonds during DNA replication. I need to use something to glue them permanently together. I'm going to need something like a DNA ligase. So DNA ligase acts as a glue. It seals the nicks and forms a phosphodiester bond here and here between these two fragments of two different sources of DNA. And by the time I am done, I, as a person, and this thing as a bacteria, can't tell where this new source and the old source came from. Oops, my circles stay. So the whole picture, and this is in your textbook as well, the process of doing this is conceptually super simple. We add an enzyme that cuts DNA at very well-known locations. We cut two different sources of DNA at the same location, just sequences. We put them together, we cool off the solution, and that allows them to anneal, to reform. And then I seal it all together with a ligase. Having those overhanging ends really makes this a little bit structurally easier. Blunt ends are possible, like I showed you on a few slides ago, but the overhanging ends makes it easier for us. We looked at HindD3 here, which is the one of the ones we're going to use this week. It has these overhanging ends that allow hydrogen bonds to really form. The problem, again, with blunt ends is that you can't form hydrogen bonds between the old and the new sequence. We're really relying on a ligase to glue them together. When I do this and I take my restriction and I add a little tiny piece of new DNA I want to insert them, I can use a vector to do that. So I don't know how familiar you are with the idea of vectors. As an entomologist, vectors are like a big thing that we talk about often. Vectors are an organism that transfers something from one place to the other, often without it getting caught up in the story. We talk a lot in entomology about mosquitoes as being really effective vectors. Mosquitoes are really good at passing disease on, but they're really not hugely impacted by the disease that they are carrying. So a mosquito that's carrying malaria or a mosquito that is carrying dengue, it's all going to be passed on to you, the host, with minimal impact on the mosquito itself. The mosquito is a vector. DNA vectors are molecules that accept and replicate DNA fragments, and I'm introducing them into a host cell. We do a series of cloning vectors. These are going to start to look really familiar in a second. When we have a really effective one, we have them where they have many different known restriction sites. So places where I could cut them, They can replicate in the host cell independent of the host chromosome. So I want a piece of DNA, maybe in a little circle, that sits inside of the host cell, but isn't part of that host cell. She is spunky today, I'm sorry. Hello. They can carry a gene that is a marker gene. It's something like antibiotic resistance, or maybe a reporter gene, something that encodes a protein that you can see, like glowing. And hey, this is exactly what we did. This is what we did in pGlow. We introduced a vector. It was already done for us at this point. The genes that we were interested in had been inserted. We had a marker gene, which was antibiotic resistance. We grew them on ampicillin plates so that we could select out the bacteria that had uptaken the plasmid. Only they could grow on the plate that had antibiotic in it. And we had a reporter gene. That was our actual pGlow gene that glowed in the dark so that we could literally see the ones that were expressing completely the gene that we were interested in. It looked like this, and you do have this in the week that we did the pGlow gene, but here you do not have to look at it, but look at it with a different set of eyes now. Look at that. There's EcoRI. There's HindD3. These are places where I can insert genes that I want using a correct restriction enzymes. This is a plasmid that we use for pGlow. It was 5,000, a little over 5,000 base pairs, meaning it had over 5,000 A's and C's and D's, T's and G's double bonded to, or hydrogen bonded to each other. It is double stranded, so there are two sides of the DNA around this. So we have an A to a T, and they're hydrogen, that's not a T, an A to a T, and they're hydrogen bonded to each other. Wow, my DNA is off today. And then this is this little circle of DNA. It's a plasmid. And I gave you the link here. This should be a permanent link in case, I don't know, you need this in 2028. But I also linked all of this in the Canvas site, so you can look at these if this is of interest to you. Remember the ORI here, this is the origin of replication. This AMP-R is the ampicillin resistance. This is the gene that allows us here, this selectable marker gene. It's resistant to ampicillin. And the reporter gene, I'll go back and forth here. The reporter gene here is this GLOW gene, GFP. In this particular case. So when we did this in the lab, this is an exercise that is really done to introduce you to the idea of cloning and bacterial vectors and the fact that genetic manipulation is possible. This is a designed bacteria and a designed plasmid that is really about doing this particular lab, which blows my mind, by the way, but it's absolutely designed to do these series of labs by the company that makes them for us. This is the sequence here, and I've written a little over it. When you look at the code in this plasmid, this is it. Also included in the stuff you can look at on Canvas, this, and we will look as we get into the last couple weeks of class, we'll start looking at how we understand this. This is the sequence of DNA that encodes what we did in lab. And it's frankly a little shorter than I would expect. It seems like it does a lot for just a little bit of 5,000 something base pairs. Plasmid vectors in bacteria are more useful than what we actually did in the pGlow lab. I want to be able to insert them with the gene of my choice. Maybe I'm inserting them with a gene for insulin so that I can use bacteria to make human insulin, or maybe I'm inserting them with the gene that I want to test in a situation where I'm not actually testing it in a human patient or an animal patient. I want to just see what happens in the gene itself. When I do this, you kind of know the story here, but I want to go over it. In order to insert them, I need to get this plasmid into the bacteria so the bacteria can use it. It can transcribe and translate this particular set of genes. I am either going to do this through heat shock. That's what we did. We exposed it to cold, and then sudden heat, and then cold again. or electroporation. This is a machine that we do not have at Camden, but we take electricity, we give them a little zzz of electricity, and it causes their pores to open so that the plasmid can move in. So I want to review just real briefly what we actually did in lab. I know we talked about this in lab and you did it with your hands, but let's look at what happened molecularly again, just so we're all on the same page. We transformed our E. coli by using heat shock method. And that takes advantage of the fact that the DNA is negative, and so the DNA of the plasmid that we want to insert is negative. That negativity here is a result of these phosphate groups. They are a little bit negative, PO4 to negative, and that this is also, for those of you, almost all of you are in chemistry, the or orgo, they would have been PO4H2. They give up these hydrogens, the protons, in solution. So this is also the source of the deoxyribonucleic acid. This is the acid part, and they're also negative. We're going to use this again in another part of this lecture. This negativity means that it can't pass through the membrane. Phospholipid bilayers are really heavily lipidy, like fatty. Those lipids are hydrophobic. That means that they push charged particles away, or things that have sides, like polar molecules, like water. Here, this negatively charged plasmid cannot get into the bacterial membrane here because of the structure of the membrane. We then put our plasmid, we rehydrated it in calcium chloride. When calcium chloride is immersed in water, it disassociates. So here, this calcium, the calcium becomes positively charged. This binds to the negative DNA here, and it negates the charge. This becomes an uncharged molecule, which lipidy things just really mix well with. And it allows this, I shouldn't have written on it. It allows this, oh no, it works. It allows this to pass through the membrane as pores form. When the pores form, we exposed it to heat. We caused the bacteria to kind of boop itself with little holes in it. And as those pores formed, the DNA could easily pass through into the cytosol of the bacteria itself. And when it did that, we then put it back in the cold. That allowed the membrane to reseal itself, and now this plasmid is inside the bacteria. We took our plasmid, we put it in a solution that allowed it to negate the charge on the DNA and the plasmid. We then exposed the bacteria to something that caused it to form holes. The DNA from the plasmid moved into the bacteria, and then we resealed the bacteria. When we did this, it can be replicated by the bacterial's replication mechanisms, starting at the origin of replication for the plasmid. Here in this one, this is the other one we're going to talk about today. This origin of replication is the place where we actually initiate replication. And it's going to follow all of the same rules that we did in bacterial replication 2 weeks ago. Here in our pGlow, we have this here where we inserted our glow-in-the-dark gene, and look at how it has all of these sort of recognizable restriction sites. Here too, you have HindE3, we have EcoRI. On this gene, or this plasmid, I see the same kind of places where we can use a lot of different kinds of restriction enzymes. This is called the multiple cloning site. The multiple cloning site is made up, it's a small area that is made up of different genetic combinations that are usable by different kinds of restriction enzymes. I use a restriction enzyme here on my plasmid, and I use the same restriction enzyme on the gene I want to insert. By having a lot of different variety options in my multiple cloning site on this plasmid, That means I can use the appropriate restriction enzyme for the gene that I am interested in, and I can insert it into this multiple cloning site kind of regardless of which restriction enzyme I'm using. I just have to use a matching one on my source and my vector. I then anneal the sticky ends together. DNA ligase binds it together, and I've inserted my gene into some area of this multiple cloning site of my DNA sequence on my vector. I cut it. This is my DNA that I'm interested in inserting. I allow it to anneal and then seal it together. I introduce my plasmid into the bacteria by either heat shock or electroporation, and now that bacteria is going to make this gene if it's complete. In reality, not in demonstration labs, but in reality, the most commonly used genes for this are not going to be a glow-in-the-dark gene, but I'm going to use the same ampicillin resistance gene and LAC-Z. We're coming up on, I will explain a little bit more about the operons on lactose metabolism in bacteria, but I think for right now, I can tell you that this gene, LAC-Z, is one that controls for managing lactose metabolism in bacteria. It takes the lactose, which is a type of sugar, when exposed to the bacteria, and it splits it into two different monomers, or two different smaller pieces of sugar. The most common way that this is used is a process called blue-white screening. We're going to have an ampicillin-resistant gene and the LAC-Z gene. This is a gene that controls for lactose metabolism. It encodes, and so I put this in parentheses. When we get to chapter 16, we'll talk more about it. But it encodes for a gene called beta-galactosidase. It's an enzyme that breaks down galactose, which is a type of sugar, into its component parts. When I grow bacteria on plates with ampicillin and a sugar called xgal, if beta-galactosidase is being made, the plate turns blue. So this I had to think through when I was first introduced to it, and this is not something I learned in high school or anything. This is something I learned in grad school. So I remember really, really thinking about this. I have, if we expose a bacteria to xgal, and the bacteria can, if the bacteria can cleave it, then the bacteria turn blue. If we grow bacteria in a solution or a plate with a lot of xgal on it, and the bacteria sorry, Alice is ruining. Oh my gosh. We're spunky today. This spring. If the xgal is in the plate and the bacteria cannot break it down, so it can't make beta-galactosidase, then they stay white. So if you cannot metabolize beta-galactosidase, you don't turn blue. Blue-white screening relies on the fact that I insert a plasmid with the LAC-Z gene that encodes for beta-galactosidase. And my cleavage site, where I am using a restriction enzyme to add in a new gene, it is in the middle of the LAC-Z gene. So if the fragment is incorporated, it breaks the LAC-Z gene in half, essentially, and the bacteria itself can no longer make the gene. What color does the bacteria turn if it can't make beta-galactosidase and I put it in a plate with xgal sugar? They grow on the plate and stay white. They can tolerate the antibiotic because we've inserted an antibiotic-resistant gene with the plasmid that we gave them. They get that no matter what. If I successfully inserted my gene that I am interested in, I inserted it breaking the LAC-Z gene, so it no longer can make beta-galactosidase, so it can't break down the XGAL sugar, so it stays white. White means it worked. Blue means it doesn't work. Or we didn't insert the plasmid successfully. So on a plate here, it's very easy for me to see who is still making beta-galactosidase and who is not making beta-galactosidase. Little bit summary, and the rest of this is pretty short. I want to talk very briefly about the idea of a genomic library. I make this by sequencing tons of different parts, little fragments of a DNA, parts of a genome. I take the cell's DNA out, I randomly cut it with a bunch of restriction enzyme, and then I insert those fragments into vectors. I then sequence those fragments. Those sequence fragments are overlapped. We will do this in a lab. We do it with a machine now. You're not going to have to do it with your eyes. This works great if you're doing something very small. If I'm overlapping genes and trying to find out what's the whole sequence and I only have 500 base pairs, this is okay. If I have the entire human genome, this is not a practical method. It uses a lot more processing power than we actually have available in computers. It also includes all the introns in this process, so it doesn't give me a functional genomic library. It gives me what is the code inside of you, but not what parts of that you are actually using. And I don't know if you remember from a long time ago, but we talked about the fact that a lot of your DNA code is actually not functional. It's doing support, it's promoters, it's doing ancient stuff that we don't access anymore. So it's interesting, but it's not particularly practical if I'm using it to understand how you access and function rather than I'm just interested in what is your secret code. Complementary DNA libraries, cDNA libraries, are a different version of creating a genomic library. I'm using mRNA to try to then backward transcribe. It's called reverse transcription. I take my mRNA that your cells are making, and then I figure out what was the DNA code from that. It's actually super cool how we do this. I think it's a good review of how we talked about DNA replication and transcription translation. When I transcribe something in a eukaryote, I go to this point at the end before it leaves the nucleus where it gets a cap and a poly A tail. So we know that every single transcript that you are exporting from the nucleus ends in which means I can grab it. Because all I need to do is make a primer, a poly T primer, or lots of T's that are just floating around in solution. And they will form hydrogen bonds with this If I insert a poly T, I'm inserting, this is DNA because it is thymine. I insert this, I can then put it in solution with a long strain of T's. I can use this as my primer to then DNA replicate, DNA complement to my mRNA transcript. I then allow these to break apart. I synthesize in fragments a complement to the DNA I made in the lab. I synthesize a complement to it, and then I use DNA ligase one more time to seal the whole process together. When I do this, I have a copy of the mRNA as DNA. This double-stranded complementary DNA is a sequence that I use inside of my body. I can then insert this into a vector. I can sequence it. If I insert a vector, if this was the code for insulin, let's say that's a bit silly, but if that's a code for insulin, I now have the DNA sequence at this point for insulin. I can insert that in using a restriction enzyme to a vector, And if it's successfully white at the end of that process, I know I can select those particular cells and use them to manufacture insulin for myself in a plate, rather than taking it from people or breeding pigs for that. So I want to move on to what other procedures we're going to do in the lab this week. We're going to start using the PCR machine. What that means is we're going to start making lots of copies of the DNA that we are working with. This process wasn't developed until the 1980s. It's a fairly new process. When I was in college, this was like the new fancy thing. I was, by the time I got to grad school, surprised at how quickly it had taken over the lab. We had them like super accessible to everyone. We have 2 in the back room. I'll show you. These machines make using DNA in research possible, because what they do is they take a really tiny piece of DNA and they amplify it. They make it so that it has lots of copies of the fragment we are interested in. And that means I can mess with it. I can get a sample from someplace and do a lot of different tests on it. I can drop half of it on the lab floor. Some of it can be contaminated without losing my original sample. And that tiny little advent in DNA technology is what really has allowed us to do all of the other things that we can do today, because we can copy DNA in the lab. In a tube, we're gonna have a tiny tube. We're gonna place the fragments we are interested in. We're gonna add some DNA polymerase. This is often still used as Taq polymerase. This is T. aquaticus. It comes from an organism that tolerates really high heat, so it's good for use in the lab for sort of brute force DNA replication in a tube. It also does not interact with the DNA in your body. So it's always good not to mess with your own DNA while you're in the lab. We are going to add a magnesium cofactor. This allows the process to have the enzymes to have the shape that they need. It's going to also speed up the reaction. And we'll add 4 types of DNA nucleotide triphosphates. These are going to be adenines, cytosines, guanines, and thymines that are loose in solution, and they still have 3 phosphate groups attached to them. They carry their own energy with them, so that they're essentially will break off 2 of those phosphates and they'll fall off as a pyrophosphate or a 2 inorganic phosphate combo. And then we'll be able to facilitate this reaction. We're also going to need some primers. We've talked about primers a lot now, but primers basically make a free 3 available end so that I can replicate my DNA following all the rules of DNA. It's about 20 nucleotides long. I buy these on the internet. I am going to proceed in 3 steps, which involves no thought on your process. You put them into the machine. If all goes well, I don't want to curse us, if all goes well, the machine is a thermocycler. It cycles through lots of different temperatures. It goes through a process of denaturation, which separates the 2 strands of DNA. It breaks the hydrogen bonds, but not the covalent bonds. We then drop the temperature where the primers are likely to anneal. These 20 fragment pieces of DNA bind hydrogen bonds to a sequence that matches them exactly. And then we raise the temperature up just a little bit so that it's optimal for the polymerase that we added, in this case Taq polymerase, to build and elongate a sequence of DNA. Every time we do this, it cycles for a short period of time. The one I had in my lab in college took hours. My understanding is ours really takes very little time compared to that. We'll run it as long as necessary. We can set it to run for 500 segments, 500 sequences. We'd have 1 billion fragments of DNA by the end. Each round, the number of fragments double. So we start out with our target sequence, we've cut it, we've used restriction enzymes to get it about the right length, but still got some fuzzy ends on the side. Our first cycle, we separate it, we add primers, and we make 2 molecules. Our second cycle, we do it again. We denature it, we anneal primers, we separate, we allow Taq polymerase to do its thing, and by the end of that, we have 4. The third cycle, we take these 4, we run them through the denaturation, annealing, extending cycle one more time, and at that point we have 8. 2, 4, 8 product molecules. When we do this, you see how these two are highlighted? Every time we do this, as we build our primer, we build our new sequence, our primer attaches, and we elongate until we reach the end of whatever fragment we cut. Our primer elongates and it reaches the end of whatever fragment it cut. Well past what our actual target sequence is, more information than we need. Every time we run through a series of cycles, because the primers are always attaching in the same place, we end up in the second cycle with one that builds on, this is one of my pieces that I made here. It's cut in the right place at the end of the fragment we are interested on one side, and it's too long on the other. When my primer binds and we build it, it reaches the end of the target sequence, and there is no more DNA here. So this one here is now just the fragment that we are interested in. This one here is just the fragment that we are interested in. As I do it a third time, this one and this one here are already just my target sequence, no fuzzy ends on the end side, so that when these ones and this one get through the third cycle, I have a piece of double-stranded DNA here that is just my fragment, a piece that is just my fragment. These ones are going to start to overwhelm my sequence, and by the time I do this 64 times or 500 times, to be absolutely ridiculous and wasteful, frankly, almost all of the solution will be only my target sequence with no fuzzy ends on either sides. I then have almost my entire sequence is all exactly the length of the fragment that I'm interested in. And now I want you to think back to the elephants. How did we identify different alleles in the elephants? I always imagine you're all shouting the answer at me, but probably not. You're probably sitting patiently waiting. They were different lengths, right? So we ran them through the imaginary electrophoresis to look at the different alleles in our samples from the tusks versus the murdered elephants, and to try to find, we were looking for which population and which individual elephants. We used electrophoresis for that. I will point out that in reality, in a modern lab, you are probably not using gel electrophoresis. I do not have access to the type of equipment that you're going to use in a real lab doing this. Electrophoresis is a good model for this, but this is a good model from the 1990s. You are probably going to use a spectrophotometer to actually do this. You'll get a more accurate sizing of them, and you'll get more information. For us, we're going to use gel electrophoresis because it's a community college, right? And I don't have access to a research lab. But we will do with the last bit of the elephants, as I bring it in, as we come back to part 2 of case 2, I want to show you what you would actually do in a modern lab. I just don't have the actual equipment for you. We're going to look at building our sequences that we made, the fragments. We're going to look at them by size. We're never going to know what the sequences are with this procedure. We're not going to know their A's and C's and G's and T's, but we will know if they are the same or if they are different. We are going to do this over the 2 days of lab. We're going to make these gels, and if all goes well, we're going to take the pieces we cut with the restriction enzymes, we're going to insert them into the wells along with our control, and we're going to turn it on. Because DNA is negative, with that negative phosphate group, it's going to move away from the negative side of the electricity. This is black towards red, which is going to be the positive. So we're going to run towards red. Now that's read. Run towards red. And then we're going to be able to visualize, look at the bands if all goes well. I have 2 different versions of how we can stain these. I'm going to show you both. There is a video posted on Canvas that I will ask you to watch at some point. He's doing it slightly differently, but he shows some of the little tiny techniques that I think are helpful to you doing this. There is, inside the machine we are going to use, and I have a couple of the machines for doing electrophoresis, so we'll be able to all look at it together. There's a tiny, tiny wire that's made out of platinum. No matter what else happens, you can't touch that wire. That wire is incredibly fragile. If you touch it and move it out of position or break some of the platinum off, this process doesn't work. So we're going to be super careful with that because these machines they look like plastic boxes, but they're $1,000 machines. We're going to be also very careful with it. And if everything has worked and they've been loved, we'll be able to get the bands. And then we'll be able to say these are same or different. Same size, different size. And that actually is enough for many procedures to give me all the information I need. Is this the elephant C, or is it elephant 10, or is it elephant 20? And that was enough in our elephant experiments to give us all the data we needed. So we return after spring break. We're going to do some DNA analysis, do restriction enzyme, and then crime scene analysis. So 2 weeks of practicing using the PCR machine, practicing the analytics. I am going to then move on in lecture. Those 2 weeks, we're going to do transcription and then translation, and then lac operon, how we actually express things, whether you're a bacteria or eukaryote. After that, I think it'll work better if we actually go to the lab itself. I'm going to try to take you to the computer lab one evening, and we're going to look at bioinformatics and how that analytics is actually done. It's awesome that some of this is just freely available on the internet. A lot of the databases and the analytics that we need to do is something you can just log in and see. I have a set of exercises doing that, but the cool part of it is you can 100% do it for the rest of your life at home by logging into those same, if you're analyzing DNA at home, by logging into those same websites and letting it do stuff for you. So I have some formulaic ones to practice through, but that I think is where we start getting into how is this actually used in a world where it's the 2020s and we're analyzing DNA with the understanding of 100 years behind us, at least. So with that, I have some practice. So practice with me or not. I have these are in your student version of this. There's no answers. I'm going to give you a second to look at this. I have a question like this in the, I pulled it out of the homework and then I put it back in and I pulled it out. I'm not sure if I left it in at this point. I think it's a great question, but it takes a little bit of fiddling. So I'll give you a second. How would you even begin to approach a question like this? So this means it weighs the same. Basically, it has the same amount of A's and T's and C's and G's. That tetranucleotide idea that the original researchers thought was going to be fundamental to DNA. So I break it every 5 bases. How often do I expect the cuts to happen? I put this here for you because I think that once you see this pattern, this is something that is actually useful to you. If the bases are present in equal proportions, so A and C is equal to T and equal to G, there's 25% of each one, the probability of any one of the 4 bases is 0.25 at any location. So if I have X, X, X is my sequence, boop, 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 boop. If I'm looking at this side of the DNA, the probability that this is an adenine is 25%. The probability that it's a thymine is 25%, and so on. The probability of at any one location is 25%. In any 5 sequences. So this is going to be the probability of any one of these, and how long is our sequence. The average fragment size in this case that we're looking for is 5 bases, so 0.25 to the 5th, which is 1,024 places on average that I expect my restriction enzyme to cut. And insert, hopefully, the DNA that I'm interested in. In a PCR chain reaction, in a PCR reaction, so polymerase chain reaction, the purpose of the step where the temperature is between 70 and 75 is, so I put the glue over here. These temperatures matter. I normally tell you, like, get the gist of them. These matter. If your temperature is off, it's going to do wonky things. You want to make sure it's actually moving through the correct temperatures to do this procedure. In this case, 70 to 75 is about the optimal temperature for the Taq polymerase that we're using, or you'll use different ones depending on your lab, but this is the optimal temperature. This temperature here of extending the primers or actually building the new sequence of DNA matters. It depends on what polymerase you are using. This is going to be written on the procedure as you do it in a lab. So you don't have to envision that. It comes in the packaging. Whoop, whoop, whoop. Which of these DNA sequences is palindromic? So a palindrome is the same forward and backwards, like mom. And now I will tell you that this is not. This would also be D. I don't know why it's called A there. You're probably like CCC and then CCC. But it's the other strand it has to be palindromic with. This is CCCC and this is GGGG. That is not a palindrome. So it has to be in the double strand. This one, ATGCAT, the opposite of it, ATGCAT. And I will have these on the quizzes this week, the homework, and probably the exam. I think for me, palindromic always tricks me up because I want it to be this, right? A to A, T to T, G to G. But this is not a palindrome because it's within one side of the DNA. We need it palindromic between the two sides. So this one would be TACCAT. This is not the opposite of this. This is not the opposite of this. And with that, that's just part 1 of chapter 20. That is all we are doing this week. In chapter 20, I have some translation for you, mostly translation. I think it's stuff you already know. I am concerned from the exam that stuff you already know is like you know it, but you're not sure, you don't know it well. So I want to review it with you in lecture lab again. I know you have done translation. Treat it as if it is a new thing before you start the video, and you've never heard of it before. I think that will do wonders for you, making sure that it's at the forefront of your mind the next time we talk about it. I will see you in the next video. Alice will see you in the next video. And I'll see you next week. All right. Bye-bye.